United States of America, for delivering the 27th lecture of IPS Platinum Jubilee Lecture Series. Now I request Dr. Barnwal Sir, National Professor, IRA New Delhi, to welcome the speaker and introduce him.
Okay, thank you, Dr. Padmeshwari. Am I audible?
Yes.
Yes,
Okay.
sir.
A very good morning to all of you, and I have a great privilege to introduce Dr. Abhijit Roy, whom I know for the last more than 22 years when he joined IRI as a master's degree student, and then he did his PhD. So currently, he is a research plant pathologist at USDA ARS, NEA BARC, and he did his graduation from Bidhan Chandra Krishi Vishwavidyalaya in 1994, followed by master's degree from IRI in 1997 and PhD in plant pathology in 2001. And then he moved to US as a postdoc fellow and worked there at Citrus Research and Education Center, Florida. Somebody is speaking. Kindly
Please mute your mic.
And after doing postdoc, he, for a brief period, moved to India and worked at Bangalore, Genie, and again moved to US as a research molecular biologist at USDA Agriculture Research Service. In fact, he has a very excellent credentials during his study at IRI. He got Professor M.J. Narasimham Academic Merit Award, which was presented by Indian Phytopathological Society. And he also got Jawaharlal Nehru Award for best, or rather outstanding postgraduate research work for his work at IRI. And then he also got Lee M. Hutchins Award for in 2019 by the American Phytopathological Society for outstanding research contribution on basic and applied research on diseases of perennial citrus fruit crops. He is also member of International Committee on Taxonomy of Viruses for the genus Bluneri virus, Silevirus, and Hygre virus study group under the family Ketaviridae. So I can read on. He has been recognized for his work as a senior editor of Plant Disease of American Phytopathological Society. And recently, he has been focusing on a lot of work on exotic viruses. And he has contributed extensively on citrus leprosis disease, on which he is going to deliver a talk today. Citrus leprosis disease is an economically important disease affecting citrus, and it is emerging and occurring in South and Central America from Argentina to Mexico. It is transmitted by mites and produces localized symptoms on different parts of citrus. So he has characterized the virus using modern high throughput sequencing. I will not come in between. I will not take more time, and I would request Dr. Abhijit Roy to deliver his talk on leprosis disease. So, Dr. Roy, Abhijit.
Yeah. Can you see?
Not yet.
No, not yet. Sure.
Just hold on. Can you see?
Blank.
Yes, some
yes.
screen has come.
The screen has come.
Okay.
Yeah,
Yes. Now you can.
now it is coming, sir.
Now it's coming.
Yeah, full slide mode. Can you put it? Yes.
Okay.
Yes.
Okay. Just one sec. Good morning, everyone. It is audible, right?
Yeah, you are audible,
Yes.
sir.
Okay. Good morning, everyone, and thank you to Indian Pathological Society for inviting me to share my research with all of you, and mainly on the citrus leprosis disease complex. Indeed, it is a great honor for me. I started work on citrus viruses, as Dr. Barnwal already told, when I was a graduate student. And there is no doubt that the knowledge and experience I gathered those years are the stepping stone in my career. And I am thankful to my chairperson, Dr. Padma Ramachandran, and the renowned citrus virologist, Dr. Y.S. Alawat, for their guidance throughout my PhD dissertation. Before I move forward to my presentation topic, I just want to give you a brief introduction about why citrus I am working, and how it is related to my current job. So after that, I'm going to give going to the topic. Okay, citrus, as everybody knows, is one of the most economically important food crops worldwide. The global citrus production in 2020-21 even has been increased 1.4 million ton, that is around 48.8 million, due to the favorable weather in the Brazil, Mexico, and Turkey. But unfortunately, in US, since last 10 years, there's production always coming down. And this year also, we have a 11% reduction in our total productions. And this is mainly due to the poor fruit setting in California and continued decline in area and yield as a result of the citrus greening or honglong bean in Florida. As the Florida is famous for mainly production of the orange juice, when you have a reduction in the yield, definitely orange juice production also come down. Even though the global orange production forecast was matched the consumption, but still in US, we have a 7% reduction as compared to the last year, and it is a record low of orange juice production this year. So, as it is citrus, but I am in Maryland, which is the far away from the citrus growing region, but still I got the responsibility to work on citrus diseases. And mainly, I'm working on the citrus exotic pathogens, and those basically for the host pathogens and vector interaction among these pathogens. And develop the uses of characterize the interaction of the high consequence of citrus pathogens and its host, and established the etiology of novel exotic citrus diseases. And I am also playing as a role for the subject matter expert and resolving the scientific problem associated with the exotic citrus pathogens, mainly for the quarantine program. Before I moved to the ARS, I was working with the Animal Plant Health Inspection Service for the plant protection quarantine. So I never miss the work on the citrus, even I am working on the exotic pathogens. So here I have another responsibility, that is basically I am working and maintaining the exotic pathogens of citrus collections. Exotic pathogens of citrus collections, and that by involving the citrus pathologists, researchers from the universities, government agencies, citrus industries, and the stakeholders. So why we are creating the history, the exotic pathogen citrus collections? And what is the exotic pathogens basically? The exotic pathogens are those which either do not occur or occur within only limited distribution in the United States. This exotic pathogens of citrus collection was created in 1984 and is basically funded by the USDA ARS. The unique facility was destroyed by a tornado in 2002, and the facility was rebuilt in November 2006, and which is around 32,000 square feet in greenhouse. Why we are maintaining the greenhouse exotic collection here, as I have mentioned, that this is just 1,500 kilometers or 930 miles away from the commercial citrus growing region that in Florida or in California. We are allowing the cooperative scientists from Florida and California, as well as the scientists who are working in citrus from the different parts of the world, who can come and work as a visiting scientist and using our collections to do their research work. So I have plenty of options for a visiting scientist who can come and work in my lab. So another role, when I am maintaining the exotic pathogen of citrus collection, I have a cooperator for National Clean Plain Network project. So basically, how we can create a disease-free plant that can be used to grow in the citrus growing area. So for that purpose, I am maintaining the exotic pathogen like citrus virus and the stem-beating strain from the different parts of the globe, world. I am having the citrus yellow mosaic virus from India, citrus chlorotic dwarf associated virus from Turkey, and I have a HLV citrus Liberobacter asiaticus and the collection for the citrus Liberobacter africanus from Africa. I am also maintaining the citrus canker pathogen, the Xylella fastidiosa and Spiroplasma citrus. The main work for us is to maintain those plant material and provide those control as requested for the assay by the NCPN lab or other universities or laboratories, and develop the new diagnostic methods for those exotic pathogens and identify or characterize if novel pathogens infect the citrus. So there is multiple ways when a foreign pathogen, there is multiple ways where the foreign pathogens disease poses a great threat to US citrus industry. Why? How? The world is becoming smaller as a major transport network continue to expand. Transportation of people and agricultural goods are increasing every year due to expansion of global transport pathogens, and their vectors can now move further, faster, and greater numbers than the before. The greater example is citrus canker. The citrus canker was exotic for Florida, but after introduction, now this were having a endemic disease and that present in Florida. So as you see, there's other foreign disease that is like citrus black spot, orange sweet orange scab, and citrus greening or honglong bean. Those are the pathogens earlier considered as exotic for the United States. But nowadays, all the pathogens are present here and it is now in domestic states. So we are thinking, so those pathogens are already here. Then what will be our next target and which pathogens can enter in the US? So if we see the list for the exotic pathogens that having in the regulatory concern for citrus diseases in United States, you can see that most of them are virus diseases, and except one, that is citrus variegated chlorosis that caused by Xylella fastidiosa. But due to advent of high throughput sequencing, there are more viruses has come, like earlier unetiology or unknown etiological diseases like citrus concave gum and citrus impetratura are now characterized. There is two more new viruses has been recorded, one from Pakistan and another from in China. like citrus yellow leaf mottle disease and citrus mosaic disease. So now we have to work on those pathogens as diseases are exotic for United States. Now moving to the citrus leprosis, as we are considering the citrus leprosis is the most important pathogen that we are looking for that can enter anytime in the United States. The citrus leprosis is basically a new world disease, and the citrus leprosis viruses are distributed in all Central American countries, most of the South American countries, except the Ecuador and Peru, where the viruses are also moved towards the northwards and found in Mexico. And the disease was earlier reported from Florida. And interestingly, the disease is disappeared after mid-1960s. So we are considering the Florida is the hot place, and there is high chance of re-emergence of citrus leprosis disease. So why citrus leprosis are considered as an emerging threat? As I have mentioned, the citrus leprosis was first reported more than 125 years before in Florida and remained there almost 100 years and suddenly disappeared. And the disease is now present in all Central American and South American, most of South American countries, and moved to Mexico. As the disease are present in Florida, we definitely have a susceptible host, and we have the virulent vector. If the pathogens is entered in Florida, Brachypulpa species, the flat mite, and the environment condition. So all that will fulfill the disease triangle, and there is a high chance of the disease development if the pathogen introduced. In 2020, citrus leprosis disease associated with one of the viruses has been reported from Hawaii. So there is a chance that virus can enter from Hawaii to the mainland of US anytime. So we have to be prepared to prevent or eradicate the virus if introduced. In 2021, when we were looking for the plants viruses, we found the similar viruses in ornamentals in Florida and orchids in California. So before I move to today's topic, I just want to give a definition about the convergent evolution. So what is convergent evolution? Convergent evolution refers to the independent evolution of similar traits from different ancestral points. As you can see, the ancestors are the different, but they're coming and joined a point where you have certain characteristics in the plants or in pathogens that are evolving that way. So that is called the convergence evolution. So basically that evolved a similar situation from a different ancestral states. The convergence often results from similar genetic changes, which can emerge the evolution of similar or identical mutations in independent lineages. The term as a parallel evolution. As you know, that insects, birds, or bats both have evolved the wings, but their ancestors are the different. So this is not only for the plant, not only for the animals or the insect. Even if you look in the plants in North American cacti and the euphorbias that commonly known as sponge in Southern Africa are evolved through convergent evolution, because the euphorbias that having the similar body shapes and structures that evolved the same like the cacti in North America, and is that you can see the spine also there present, but it is very distantly related than cacti. So these are the evolution coming through convergence. So here, citrus leprosis that I am going to talk today is a disease complex. Why it is disease complex? Because it is caused by two unrelated taxa of viruses. One of the virus That is where you can see the short bacilliform virions inside the inside the endoplasmic reticulum in the cytoplasm. Whereas in the other one is the nuclear type, where you can see the bullet-shaped particle inside the most of the time inside the nucleus, and sometimes you can see even in the cytoplasm. And both of them are produce almost similar kind of symptoms in the leaves, fruits, and the bud. But then what is the similarity between these two pathogens? The similarity is there that both the pathogens, both the viruses are transmitted by flat mite, the brevipulpus species. So what is there is multiple species of the brevipulpus that are involved in the virus transmission that I'm going to talk in the future slides. So brevipulpus species is a very small mites, flat mites, is 200 to 400 nanometer in the length, is a red in color and polyphagous in nature. They belong to the genus Brevipulpus and serve as a vector for both cytoplasmic and nuclear leprosy viruses. There are 5 major species of Brevipulpus like Californicus, Phoenicis, Papayensis, Yorthosi, and Ovovetus are associated mainly with the citrus leprosis transmission. They generally produce the localized infection and the similar symptoms in different citrus species, but there is no systemic host has been identified. That is very important, that even this virus having infecting, producing the localized infection, but there is no systemic host till now has been identified. So Genome Organization, the Brevipulpus transmitted viruses might be a great example of convergent evolution of plant viruses. The convergent evolution occurs when 2 different taxa of viruses, those live inside the mite vector Brevipulpus species, become more alike in appearance, size, and produce similar symptoms in the host plant. In this case, they select the similar adaptation from 2 unrelated taxa of viruses. Here you can see the multiple viruses that belongs to either Cilivirus genus or Dichoroviruses. Those are transmitted by the Brevipulpus species. And among these, only citrus are infected by both Cilivirus, that is the cytoplasmic type, and the Dichorovirus, that is the nuclear type. So, outline of my today's presentation will be based on the taxonomy of the Brevipulpus transmitted viruses, those infecting citrus, their genome organization, followed by what kind of symptoms they are produced, the characteristics of those virus particles, how taxonomically we can identify the Brevipulpus species, those are involved in virus transmission, and their phylogeny. So till today, there is more than 6 viruses and their 7 different strains are involved in citrus leprosis disease production. The citrus leprosis, here you can see there's 2 groups of viruses. One cytoplasmic type belongs to the family Chaetophyridae, and another group of that is the nuclear type that belongs to the Leprosy. Both the viruses are in the bipartite in nature and is transmitted by Brevipulpus mite. All the details of each of the families I am going to present individually. So with the advent of high throughput sequencing, that many viruses came in the limelight within the last 10 years. Starting with the family Chaetophyridae. Chaetophyridae is a brand new, as you know, I am the member, one of the members who created that name Chaetophyridae. The Chaetophyridae is consisted of 3 genus. That is the Brunner virus, Higiri virus, and the Cili virus. we are mainly interested about the silly-viruses and higgly-viruses because these two viruses are causing or disease like leprosis-like disease in citrus. So in the silly-viruses, we have a 3 species, that is citrus leprosy virus C, C2, and very recently added another virus for the passion fruit green spot virus. Till now, the passion fruit green spot virus is not infecting citrus. So basically, we are going to talk about these 2 viruses today. How it's looked like their genome organization. If you see the genome for the silly-viruses and the higgly-viruses, the silly-viruses are the bipartite, higgly-viruses are tripartite, where HESB RNA1 is almost look like same like the silly-viruses. Silly-viruses contain 2 ORF. One is mainly the big polyprotein, the P285, and another is putative coat protein. The RNA2 is having the conserved domain, conserved ORF for the glycoprotein-like structure, the P61, movement protein P32, and the insect membrane-specific feeding the insect protein like P24, and the most variable protein like P15. The interesting part for the silly-virus is there is a big intergenic region in between ORF1 and ORF2. So now moving to the family Rhabdophilidae. Why I am telling 2 family together, so that you can get an idea what is the similarity or dissimilarity between these 2 kind of viruses and why they are convergently evolved. The family Rhabdophilidae are ecologically diverse and very old family, and that includes the viruses that infecting plant, animals, that also include the mammals, birds, reptiles, and fish. Rhabdoviruses infecting the plant, only the higher plants. Rhabdovirus is also detected in the invertebrate and arthropod. The family Rhabdophilidae almost having the 40 genera, and that had been reclassified again the last year, that concised into 20 genera, where 144 species already included, and rest of the 102 species are still unclassified. So here, we are mainly going to talk about the subfamily Beta Rhabdophilidae, where those are the viruses on this family, subfamily that infecting the higher plants. So here, Beta Rhabdophilidae also having the 6 genus, the Cytorhabdovirus, and the Nucleorhabdovirus is further divided into alpha, beta, and gamma Nucleorhabdoviruses. Interesting thing, if you come in the genome segment, you can see the Cytorhabdoviruses is totally unsegmented, where the Nucleorhabdoviruses can be unsegmented or bisegmented. And when the bisegmented, this is the genus belongs to the Dichoroviruses that identified very recently in 2006, and the genus name was approved in 2017. So this is the only genus in the Rhabdophilidae family who having the bisegmented RNA along with the Varicosivirus. But here, the Dichoroviruses infecting the citrus, we are mainly interested. Another interesting part, if you see, the vector that is associated with the Dichoroviruses is the flat mites, where the Cytorhabdo and other Nucleorhabdoviruses that is unsegmented is transmitted by the different vectors, like aphids, plant hopper, leaf hoppers, or even whitefly. So for your information or for idea, if you look how it's looked, the genome organization for all the rhabdoviruses that infecting the plants. Mainly the unsegmented genome that is starting with the leader to trailer sequence, that is the nucleocapsid, phosphoprotein, movement protein, the matrix protein, and glycoprotein, and followed by the large polyprotein that encoded the RDRP region. And if you count by the Dichoroviruses, those are having the same ORF, but it has been split into the two segments where L protein is coming on the RNA too. And the size of the nuclear of the virus is almost 12 to 15 kb in length. As compared to the silly viruses, that is also 13 kb, around 13 kb, 13 to 14 kb in length. Here, this is the few dicoroviruses that species has been established in last 5 years. The orchid plague virus is the type member, and other two viruses that infecting the citrus apart from the orchid plague virus is citrus leprosis virus N and citrus chlorotic spot virus. But more viruses are coming as the high throughput sequencing technology has been using, and the people are working on the brain purpose transmitted viruses. The second part, we are moving about the symptomatology, how the symptoms for the silly viruses and dicoroviruses are looked. In the silly virus, citrus leprosis symptom, citrus leprosis sample has symptoms for chlorotic leaf spot with a yellow halo. Raised brown rings were observed in the twigs or the branches. Brown ring spot with a central depression were common symptoms on fruit. In advanced stages, those are the coalesced and creating a brown tissue and it becoming hard. When it goes to the passion fruit, the similar kind of also you can see. For when the citrus leprosis virus goes to passion fruit, it producing the chlorotic spot in the young leaves or in the senescence in the mature leaf. The passion fruit green spot virus, when identified, it also produces similar kind of symptoms like necrotic spots on the fruits and chlorotic and necrotic lesions on the leaves. While in case of the dicoroviruses, you can see dicorovirus can infect the monocot as well as dicot, that leading to the appearance of the local chlorotic or the chlorotic spots on leaves, fruits. But in one cases, experimentally, when they shows they increase the temperature of the coffee plant, coffee leaf spot virus, it can infect systematically in the experimental host. Here you can see the different symptoms by the coffee leaf spot, chlorindum chlorotic spot virus, citrus chlorotic spot virus in hibiscus, beech hibiscus, citrus leprosis virus N on fruits, orchid plague virus in different orchids, and Lidope, the monkey grass, and Smilax and the cast iron plant is recently identified from Florida, infected with the orchid plague virus. That is one of the pathogens for citrus leprosis. Now, if you see, look closely, the symptoms caused by different dicoroviruses in citrus. As you know, as I told you earlier, that orchid plague viruses has 4 different strains. That 2 of them are infecting orchids and 2 of them infecting citrus. But very recently, both the orchid strain also identified in citrus and that causing citrus leprosis symptoms. Orchid plague virus, orchid strain 1 infecting citrus, producing leprosy symptoms in South Africa, and orchid plague virus, orchid 2 strain infecting citrus in Hawaii. And we identified the citrus strain 1 and 2 from Mexico. But still, if you can see, there is a symptoms difference between for the dicorovirus infection in different citrus hosts. Most bright symptoms, the brown spot with yellow halo, you can see in the mandarin. Similar symptoms that you can see caused by the citrus leprosis virus N in Brazil. The most different symptoms you can see when the citrus is infected by citrus chlorotic spot dicoroviruses. The big yellow spots, chlorotic spots was developed in advanced stage, but this kind of spot is not as bright as infected by the dicoroviruses, orchid plague virus in citrus in Mexico, or CIUVN in Brazil. So from there, the next part, I'm going to talk about the virus particle. The virus particle moving to the first for the ciliviruses. They're producing the short bacilliform virions around 40 to 50 nanometer into 100 to 110 nanometer in length, were observed in the, as I mentioned, the endoplasmic reticulum in the cytoplasm. This is for the C, cytoplasmic C, and for the cytoplasmic C2. Both are that you can see. And even for the passion fruit green spot virus also you can see in the under the endoplasmic reticulum. So, but in case of the nuclear leprosis virus, when you do the ultra-thin section of the infected sample from different citrus and different hosts, and you can see the bullet-shaped virion that also similar size of the ciliviruses and observed in the nucleus. Most of the time you can see in the nucleus, but sometimes you can also see the particles in the cytoplasm in the form of a spoke wheel structure. So this is the characteristics of the dichoroviruses when you are finding in cytoplasm, and when you can see this transverse section and the typical rod, the nail head shape, you can see in the nucleus. Even though both the viruses almost in the similar size and shape also, but located in the different place in the plant cell. Now the most important part for this presentation related, because how we can identify the taxonomy of the which species are involved in the transmission of the ciliviruses and dichoroviruses. I am very fortunate to have a great collaborators in my team who are the world-known taxonomists from USGA ARS, in one of my colleagues, and the persons from Brazil and some person from the Australia are helping to open the chapter or open the more details about the Braver plasmide identification. So basically you can see the dorsal CT here, then the solendia on the legs. This is arrow you can see. The morphology of the cuticular plate, the spermatheca, and the distinct dorsal reticulation pattern between those species will help to identify what kind of mites we are looking for. In 2015, when we started our work, and we tried to simplify how we can classify those main species of the brevipulpus who are transmitting the cilivirus and dichorovirus. As main part, I am going to, instead of reading in the table, I can show in the pictures and let you know a story. The 2013, when we identified the second species of cilivirus CLVC2 from Colombia, then we don't know actually what species of brevipulpus is actually involved in the transmission. We did the transmission study. We feed the healthy brevipulpus mite, that is we are thinking about that time about the brevipulpus species, feed on the plant and the transfer to the healthy plants after 24 and 48 hours. And the symptoms was produced after 25 days of the inoculation, and it becomes bigger. And after 120 days, you can see the bigger form. But interestingly, we published the paper and we told that the brevipulpus species is responsible for transmission of Citrus leprosis virus C2, as same as Citrus leprosis virus C published earlier from Brazil. But after, when we joined the team with the taxonomists from the acarologists, though they are using the low temperature scanning electron microscopy to identify the what kind of brevipulpus species we have. So to confirm the whatever the species we have for the cytoplasmic transmission, we check that and we compare. This is our species that we use for the transmission, and we use two of the type species, Brevipulpus californicus and Brevipulpus species, side by side. Brevipulpus californicus that having a dorsal opisthomal CT F2 here, that is absent in case of the any other Brevipulpus species. So based on that, when we check this F2, the CT is absent. So we discarded the Babylophus californicus as one of the vector for cilivirus transmission. Then we move to the cuticular microplate pattern. If you see the microplate pattern here, the absence of microplate pattern, if you see, the cuticular microplate pattern identified species, the Babylophus finesis, because all the line, it is irregular, even it is dorsally longitudinal, but it's irregular. But you can see in the Babylophus, where you can see the separate rounded plates with a series of parallel ridge dorsally were observed as compared to the irregular microcuticular plate for the finesis. So we are discarding the finesis as a vector for the cytoplasmic C2. So we confirmed the Babylophus eorthosi. So Babylophus eorthosi is the vector for citrus leprosis virus C2 as well as C transmission. So for dicorovirus transmission, till now, the four species was identified: one Californicus, eorthosi, finesis, and papaensis. So all the orchid plague viruses are transmitting by Babylophus californicus, as those viruses having the CT of this morphology, CT F2 is present, and the spermatheca vesicle morphology as well as cuticular pattern is totally distinct from the others. Same way, Babylophus finesis was identified as a vector for citrus leprosis virus N from Brazil, and Babylophus papaensis has been identified as a vector for coffee bean spot. But Babylophus eorthosi also play a role for transmission of one of the dicorovirus transmission. As you remember, one of the symptoms I showed is a little bit larger in dicorovirus transmission, that is citrus chlorotic spot virus, that is transmitted by Babylophus eorthosi. So here is the list. You can see this is the known dicoroviruses, and these are the vectors that I have mentioned earlier. Those are transmitting the dicoroviruses. So dicorovirus and cilivirus transmission, if you look, both are transmitted by the Babylophus mite, and they are circulative and persistent in nature. But only one thing, till now, the cilivirus transmission is known as non-propagative in the mite vector, whereas nuclear type viruses are propagative in the mite vector. But this is based on the electron microscopy and what about the knowledge they have earlier. But when we started our work and we used a method that is the complementary strand specific RT-PCR, means if the cilivirus is the positive sense, we are designing the primer based on the negative sense, and when dicorovirus is the negative sense, we are designing the primer in the positive sense and trying to get a amplicon from the mite. That is the if viruses always make the complementary template during the replication. This complementary strand only found in the replicating virus population, but not inside the virion. So the replication, when the PCR we have done, we have found the typical positive strand for citrus leprosis virus N, means the orchid plague virus, dicorovirus type, and negative strand positive from the cilivirus type inside the mite. That clearly indicate that both ciliviruses and the dicoroviruses are amplifying, replicating inside the mite. So both are doing the circulative, propagative, and persistent transmission. But still, there is a controversy is going on, as the electron microscopy still not found any viroplasm inside the mite cell for the ciliviruses. That is the main characters to prove the replication inside the mite. So still it is need further study to confirm either circulating, either the ciliviruses are propagative inside the brachypulpous mite or not. So here, next we move to the phylogenetic relationship. If you see the phylogenetic relationship, the silly viruses, when we're taking the RTRP region, the silly viruses are separated completely with the Negi viruses, Bruneri viruses, and Higiri viruses with other plant viruses. So this means silly viruses are more close to the insect-borne viruses than the plant viruses. That suggested that we hypothesize that silly viruses might be originated from the insect, where they are using citrus as a secondary host for their mite-to-mite transmission. When we look for analyze the other gene coded encodes the each warhead, we found a gene encoded the P24 protein. It is more closely related to the insect-borne viruses, leggy viruses, than the plant viruses, other plant viruses, because other plant viruses do not have any P24 protein. But the citrus leprosis virus, the silly viruses or Higiri viruses, both have similar kind of protein that are responsible, that came from insect origin. So further study that we are continuously doing, trying to look for is more, how many more viruses or the strains are still coming for the silly viruses from the mite. So data sets grouping from the silly virus sequences that we generated by the high throughput sequencing from the countries like Colombia and Mexico, and those retrieved the gene bank sequences, the phylogenetic analysis of RNA2 as well as RNA1 also depicted the three different clades. That is the three different species that belongs to the silly virus. One is citrus leprosis virus C2, passion fruit bean spot virus, and citrus leprosis virus C. And again, if you see, all of them having a different subclades divided into three different subclades for citrus leprosis virus C as well as citrus leprosis virus C2. So both of them are having three different strains. So you can see how much variation you can having in each viruses, either in the species or in the strain level. So now the questions come: what kind of strategy the viruses are using to creating the new strains or new viruses coming? Obviously, the data, the analysis done by the Brazilian group on the silly viruses, but specifically in the citrus leprosis virus C, they found the recombination event is the main part that creating the new virus strain. As you can see here, the multicolored horizontal bar that representing the genome of the recombinant molecules. And major and minor parents are indicating the black and gray bars. And dashed vertical line that is the indicating the recombination breakpoint. And here, in the both the cases, you can see the recombination breakpoint. When they're using the program that is the recombination detection program, they have included the 7 different program also. When it is detected by at least 4, it considered as a recombinant event. So there is some cases, only 2 events where all the 7 programs detected this is a recombination. Mainly, one is for the intergenic region between P15 and P61, and another is the 30 nucleotide before of the 3' end. And in both the cases, the minor parent was one of the strain that is called CRT, but the major parent is not yet determined. So moving to the dichorovirus situation, where in the dichoroviruses, as I mentioned, this is infecting plant, animals, and the insect, and that's having monopartite as well as in the bipartite. So all the monopartite, either in the cytorhabdovirus or nucleorhabdovirus or bipartite, is grouped together than the viruses infecting by the animal viruses infecting the animals. So here, just moving to the dichoroviruses with the vectors, if I use the nucleocapsid protein, and constructed a phylogenetic tree. The phylogenetic relationship among dichoroviruses is established clearly, and it's differentiated two groups of dichoroviruses that infect in citrus. One group consists of all the orchid plague virus and their strain that is reported from Mexico and Colombia, and another group of dichoroviruses that is citrus leprosy virus N and citrus chlorotic spot virus that is reported from the Brazil. That means there is two different origin of viruses from two different vectors. And if you look for the vectors, vectors are also different. One side in the orchid plague virus transmitted by Californicus, where all the nuclear leprosy viruses in Brazil transmitted by Viburnus finicis. So definitely vectors play a major role in the evolution of these viruses. Interestingly, for the dichorovirus, how it is going to create the new strain. You can see when you compare the full genome sequence for the dichoroviruses, for RNA virus, RNA1 genome, you can clearly see the four subgroups, where in the RNA2, that all the subgroups 1 and 2 combine together in one clade, and 3 and 4 combine together in another clade. That means there is clear cut that one segment of the genome is reassert with the genome 1 and create a new strain for orchid plague virus dichoroviruses. The analysis showed, here you can see clearly there is an orchid plague virus is using as a parent citrus 1 and it is creating just two different parents. And you can see that even the citrus leprosy virus N created as a recombinant point, where the reassortment created the new strain for infecting citrus or in orchid plague virus. So I give you a lot of information. So just give a recap of what I mentioned about this convergent evolution of citrus leprosy viruses. The cilivirus and dichoroviruses, one is stability. The difference between these two and similarity, positive sense, dichorovirus is negative sense,
and Dr.
cilivirus
Wagner, please
is a
mute.
positive sense, dichorovirus are negative sense, but it's located in the cytoplasm. This dichorovirus is located in the nucleus. RNA1 and RNA2 both are in the same size, 9 kb and 5 kb, where it's 7 kb and 6 kb, having coding more than 5 to 7 ORF. Replicated cilivirus inside the plant cell, maybe or may not be inside the mite, but both nuclear aptoviruses are replicated inside the mite as well as inside the plant cell. Symptoms are similar, both the cases, but more severe in case of the food for the cilivirus infection. Vectors are eorthocyte, Viburnus eorthocyte. Here there's 3 viruses, Californicus, Finicis, and eorthocytes transmit the dichoroviruses. We have 3 species, and each 2 of the species have 3 different strains. And here orchid plague virus also having 4 strains and other 3 viruses. Among them, one of the viruses, citrus leprosy virus N0, that is reported from Florida, no more exists in the nature. And both the viruses using the recombination as a strategy for the virus evolution. Dichorovirus is showing the reassortment strategy for the evolution. And now we are considering if cilivirus is also having a strategy for the reassortment to develop, evolve a new virus. So connecting link. Now we have to find a connecting link between this convergent evolution. This is the citrus leprosy, citrus chlorotic spot virus, where you can see the particle in the nucleus. Citrus leprosy virus C or C2, where you can see the particles in the cytoplasm.
Sorry,
Both the
Dr.
viruses
Wagner, please
are transmitted by the eorthocyte. Both the viruses are transmitted by the Viburnus eorthocyte.
Dr. Wagner, please mute yourself.
Sir, sorry, so much sorry.
So the connecting, you can see the species. We can say the cilivirus except for the worldwide distributed of Orchid plague virus, all of the brain-purpose transmitted viruses have been reported solely in America, wherein the brain-purpose transmitted silly viruses, cytoplasmic viruses, are found only in the American continent. Therefore, the New World is likely a scenario where the origin and evolution of these brain-purpose transmitted viruses has been occurred. In conclusion, we can confirm that the convergent evolution occurs when two different taxa of viruses, those live inside the flat mite vectors, becomes more alike in appearance, size, and produce similar symptoms on host plant. In this case, environment select the similar adaptation in two unrelated taxa, Sillivirus and Dichorovirus here. They are showing the interspecific in the taxonomic level and who adapted the citrus as a secondary host by producing the localized chlorotic localizations. And this is a clear example of a parallel convergent evolution. We do not know how many genes are involved, but we are expecting multiple genes are involved in this convergent evolution. More work has to be needed to confirm this theory. So here is a couple of publications that we published to reach this conclusion. So if you need, you can go through and look for those publications in details. And this is the specific publication where we provide the hypothesis that this is a insect virus. It's originated in insect and using the citrus as a secondary host, and come out with all the taxonomy or mite related to virus transmission. And current project that we are on citrus leprosy that going in our lab, we are trying to, as you know, we have multiple viruses already in this complex. So definitely a detection method is needed. We are looking for multiplex conventional and qRT-PCR method. We are optimized the high throughput sequencing for detection of the new virus or strain in the citrus leprosy complex. We are using the high throughput sequencing data for electronic probe development and EDNA, and that is almost ongoing, and it will come, the publication will come very soon, how we can detect using the electronic probe for the virus detection. And we have trying to identify the novel host apart from the citrus where those viruses are infecting from different countries like Mexico and in Colombia. We are looking for the recombinant sillivirus that come out, that is evolved through the reassortment. And we are doing the transmission study where the virus is going to the hibiscus, either it is potential to move to citrus or not, as these viruses are present in Florida. So here, all of my collaborators, without their help, this work cannot be completed. I'm thankful to all my collaborators, nationally from the Oklahoma State University, University of Hawaii, University of Florida, University of California, University of Texas, and of course our colleague from the USDA ARS. And internationally, we have a big team from Colombia, South Africa, China, definitely from Mexico, Brazil, and Costa Rica, and Australia for the taxonomy. And we are having a collaboration, trying to establish a more collaborative with our Citrus Institute in Nagpur, in India. And no work can be completed without your funds. So if you have no funds, no work. So definitely we are thankful to funding agency, the USDA ARS and Citrus Research Development Foundation, Citrus Research Board. And thank you all for your time and attention. And I'll be happy to take your questions. Thank you.
Thanks, sir, for your wonderful and mind-provoking lecture on citrus leprosis disease complex, a classical example for convergent of plant viruses. Now, I request participants for a question session. Questions are invited from the participants.
Yes. Can I ask? This is DK Barnwal.
Sure.
Abhijit, let me first congratulate you for a very exhaustive and nice presentation on this complex virus disease. I was just wondering that you talked about to Virus and Dichrovirus. So these flat mites, do they transmit these viruses separately, or there is an example where both the viruses can be transmitted together by these flat mites?
Till now, there is no report for the viruses and dichroviruses transmitted together.
Oh.
So, and we are trying to put an experimental conditions for those species and to look if it is possible to identify. But in my opinion, when we are trying to looking for the new host to trying to see, we found a couple of plants that infecting with the different viruses and the dichroviruses together. But those are the plant not yet characterized, neither the vector has been characterized yet. So definitely in nature, there is some point you will find one of the vectors or few of the vectors transmitted both the viruses.
Okay.
And that will help us to chalk out the point for the starting for the.
One more question because these viruses produce only local infection in most of the crops. So that means this virus survives only through vectors and there is no crop plants where it produces systemic infection naturally. You said experimentally it produces systemic infection in coffee, in experimental plant.
Yeah.
But what about natural systemic infection if any plant is reported? There is a possibility which we have not yet noticed.
See, Dr. Kitajima from Brazil, he is a pioneer for this variable pulse transmitted viruses work. And that's why you can see the family name came as a Kitaviridae. We are honored his work and we give his name for the family to recognize him. So he tested more than 200 plant species and where he found some of the plant species infected with the infection, but all are the localized. See, he didn't get any kind of infection for the systemic infection. Okay. If the virus are originated in the plant, they are using the plant as a secondary host. We are not expecting to find any systemic infecting plant.
So that means vectors are the major source of carrier kind of thing. Original, they are the, first source of infection rather than secondary.
Yes.
Yeah.
Yes.
So that is a unique kind of situation for plant viruses.
Yeah, but interestingly,
so that is why we call them insect viruses rather than plant viruses.
Yes, but interestingly, both the viruses are replicating inside the plant cell also.
Oh, that's right.
So time being, so they may establish more severity or they will change. They will may combine with other viruses, create the new one to create the more infection.
Anyway, congratulations for a wonderful talk. Thank you.
Thank you, Dr. War.
Hi, Avijit. This is Dr. Das from Citrus Institute, Nagpur. Am I audible?
Yes.
So, first of all, congratulations for a very nice and informative talk. Just I was wondering about the symptom slides which you are showing. So, is there any specific diagnostic symptoms or rather a diagnostic symptoms key by which we can identify, okay, this is leprosis, this is not canker, or this is not scab, or this is not any other things? And my next question is there any specific temperature requirement for this expression of these typical symptoms? Because many times we go for survey and we try to see some of the symptoms, but most of this leaf spotting and fruit spotting symptoms, many citrus diseases are quite overlapping. So I was just wondering, is there anything specific to this leprosis virus symptom expression?
This symptom expression you can see in most of the citrus plants. It's look like depends on in the stage of infection.
Okay.
If it is in infection in the preliminary stage, probably you confuse with the other symptoms. But if it is in the mature stage, you can easily identify the leprosis like symptoms. Because your yellow halos, green spot with the yellow halos, it turns to the green brown color spot. And instead of looking for the front side of the leaf, if you just look for the back side of the leaf, you can also see the clear symptoms there. Generally, the early stage, you cannot see that, but you can see similar kind of mite infection symptom.
Absolutely.
Because citrus leprosis symptoms, mite toxin play a major role to produce similar symptom. And people can confuse it is infected with the mite or leprosis.
Right, right. Okay, thank you.
Thank you, Dr. Abhijit Roy. So it was such a comprehensive lecture on citrus leprosis virus. I'm wondering that is it another disease which farmers of US or around the globe has to worry apart from Candidatus liberobacter, which these days it's making havoc everywhere. So what is the enormity of the disease and what is the potential threat as it is there with the Candidatus liberobacter?
See, the two diseases are totally different because citrus leprosis, it is a localized. But if the disease is introduced, your fresh fruit market is come down is a zero.
Oh.
If you look in the Brazil, they are mostly affected by citrus leprosis, and they spend more than $80 million for just control for the berry purpose mite. Because in Brazil, the most of the citrus go for the juice production. If and as well as the fresh fruit market also. So, but if the plant fruit is infected, the quality of the juice is coming very poor, and the taste is bad. So that way, it is going to affect the market immediately. But but the cost, yeah, best part is that it can control. You can control the mite, but the cost of acaricide or the miticide is very high. So it is better to prevent from the beginning if you are live for a long time, and it's moved to, as the mite is a polyphagous in nature, virus can move and stay in other host.
Right. Just I have a cross question with Dr. Ashish Das. Do we have such infection over here in India, or as Dr. Roy said that it is localized?
No. Till date, leprosis has not been reported from this country, but brevipulpus mite is there very much here. So, I mean, maybe we can have to be more alert and to see,
Yes.
I mean, try to see if there any leprosis infection in any of the commercial citrus species.
Yes. Do we have such kind of quarantine at quarantine places to which help us to diagnose such kind of, like, do we import California orange or kind of oranges or something from there?
See, one thing is that this is a new world disease, and brevipulpus virus, transmitted viruses originated in Americas, mainly in the South America. So, except the orchid plague virus, this is a part of the dicotoviruses, is not reported apart from the American in any South American countries. So, orchid plague virus is distributed even in Australia, in Japan, and everywhere. And orchid is transported from one country to another country and is propagating material. And when the mites is infected, this is you cannot see in the naked eye. So if mites are there and it's moving from one country to another country, it may cause the disease. So that's the way the mite moves to the virus reported earlier from South Africa 10 years before, and the 2 years before it has been reported moved to the citrus and causing leprosis. So if mites are there, mite transmitted virus will be definitely there. Even the species are different, doesn't matter. It definitely they're having a viruses, and that is transmitting the diseases in the wild host or in the crop plant. We are not familiar that these viruses are there. People are not working on that. But if people start working on the brevipulpus viruses, transmitted viruses, definitely you will find some other viruses that may be not the exact virus, but it's related to this virus.
Yes.
Yeah. Nice, thank you. Any more questions?
Yeah, Dr. Abhijit.
Yes, Dr. Pan.
Congratulations for your wonderful lecture.
Thank you.
Actually, I have also reported it is not published that rhabdovirus-like particles on the orchids in many species of orchid from Sikkim and Darjeeling.
Yeah.
So these are supposed to be the orchid flag virus, but it is not characterized yet.
Mhm.
So how it this may play a role in the this transmission of this particular leprosis disease? It is how they are different from leprosis,
This
orchid
is,
flag virus.
see, the orchid plague, it is called orchid plague virus because it is first reported from the orchid.
Yes.
Now it is the orchid plague virus causing leprosy, so it is creating a lot of confusion in terms of regulatory point of view for the other reader who doesn't have that much of depth knowledge about the orchid plague virus. So maybe in the taxonomy in future they are going to change some kind of name change for this orchid plague virus, so people know this virus is causing the disease. But now orchid plague virus, originally found in orchid, also started infection in the citrus. So I don't know how they are going to reclassify the name, as the original virus was first described in orchid. But orchid plague virus now means if any kind of orchid plague virus, definitely it goes to citrus. There's no questions that it is experimentally proved, and it is naturally happened. In last year, it happened in Hawaii. In Hawaii, orchid plague virus was reported most 10 years before probably, and now they're having the disease.
Okay,
so if you have the right vector, right pathogen.
Vector studies actually I have not done, but only I have seen in electron microscope a lot of rhabdovirus-like particles.
That is high chance, because Sikkim is a place where they are having a lot of orchids. They're growing and they're importing the orchids from the different countries. So there is a high chance that, because that orchid plague virus is almost reported most of the countries.
Yes. But it is in our country, it is in many species I have seen in the electron microscope only. We tried in the PCR, but we could not get the result.
But one important thing is orchid plague virus. Their introduction of orchid plague virus in different is the same. So either you're collecting the sample from Japan or in Brazil or Australia, the sequence similarity will come almost 98 to 99%. That much of you can see. So if they are changing, that changing is in RNA1 in the genome. RNA2 is remain the constant. The Ricoroviruses, whatever till now has been changes, the changes in the genome in the RNA1. So RNA2 never changed. So it is a very unusual thing, but it is very concept. So you will not see that much of variation in the sequence until you have a totally new virus strain coming. But it will go to other.
Okay, thank you.
Thank you. Anybody have any more questions to ask? Dr. Parmeshwari?
Thank you, sir. Now I request Dr. Prasad Jambulkar to give a vote of thanks.
Okay, thank you, Dr. Parmeshwari. So it was such an excellent and comprehensive presentation, and thanks, Dr. Abhijit Roy. You have introduced us, all of us, with a very exhaustive and a new virus, which has a potential threat to the citrus industry as well as the citrus orchard. I am very much grateful that you have spared your time, and that too in odd hours from your location. So we are very much grateful to you for this gesture. And at the same time, I must thank to our Dr. Dilip Lakshman sir, who has introduced you to all of us. See, I must say you don't need introduction, because you are our super senior. I was also part of IRI, Plant Pathology Department, and we used to hear about you from Dr. Padma Ramchandra ma'am, and it's nice to hear from you. Thank you, Dr. Lakshman, for all your support to conduct this talk series.
Thank you, Prashant. And Prashant, I consider him as my student. So I also had a PhD in plant virology. So,
Yeah.
so my career got distracted. So I switched to fungal pathogens. But I worked a lot on double-stranded RNAs, endonaviruses, and also shrimp viruses in between. So I enjoyed Avijit's talk and also the discussion that went with it. Thank you.
Thank you, sir. I will also thank to Dr. B.N. Chakravarti, sir, Dr. Barnwal, sir, for sparing their valuable time for this lecture. Also, Dr. Pant, sir.
Thank you.
Thank you. Thank you very much. Thank you, Dr. Roy. So see
Thank
you
you.
sometime. Make a chance to come to here in IPS conferences and all, so that we can
Sure,
interact
sure.
more.
Yeah, no, I'm going to give the thanks to Dr. Dilip Lakshmans because he's the person who gave me the opportunity to connected you guys to give a talk. And thank you to see several new known faces that I left 20 years before, to all of my seniors and who helps and a lot of things in here. I can see by name, those people are here, and I'm thank you very much. And in future, if you need any help or any kind of collaborative work, interested to work on citrus viruses, you are welcome to work.
Thank you.
Welcome to come and work in our place.
Thank you. Thank you, sir.
Thank you, sir. Thank you.
Thank you.
Thank you, Dr. Abhijit.
Thank you, Dr. Bag. Thanks a lot.
Okay, then. Bye-bye.
Bye-bye.
So good night from
Good
here.
night. Good
I
night,
think
sir. Good
Abhijit
night
could
to
have a
both
good
of you.
sleep now.
Good night to both of you. Thank you, sir.
Okay.
What is the time at the US now, Dr. Roy?
1:20.
Oh, 1:10.
Yeah, they are joining us in odd hours. So we are grateful to you. Thank you. Okay, sir.
Okay, thank you.
Okay, thank you. Thank you.
Thank
Okay.
you. Bye-bye.
Nice to see you.